This is the pulp-stick sample preparation kit from Duckybio, a menu preparation kit. Now, let's open the kit. Which contains spending solution, wash solution, illusion buffer, protein key, and magnetic beads. This is the magnetic stand that will be used in the experiment. It has a magnetic plate inside that can be removed from the magnetic stand, and there is an arrow indicating the direction of the magnetic plate. The experiment also requires a microcentrifuge, a vortex, a block heater, a timer, a miniature bench vacuum pump, a certain number of centrifuge tubes, some pipettes, and aerosol-resistant micropipette tips. In particular, the 200 microliters non-filtered pipette tips are required with a vacuum pump to remove the supernatant. Next, we start the sample preparation. Here, we take the workflow A to workflow D of one test sample as an example for demonstration. First, set a block heater to 37 degrees centigrade. Remove the magnetic plate from the magnetic stand and place 4 centrifugal tubes on the hose of the magnetic stand and mark them. Keep positive and negative samples separate as much as possible. Vortex the magnetic beads thoroughly until the color of the bottom is uniform and there is no magnetic beads sediment. Add 10 microliters of magnetic bead to each tube. Vortex the protein key. Add 10 microliters to each tube. The 600 microliters and in solution to each tube. Vortex the internal positive control. Add 10 microliters to each tube. The internal positive control offered by residual DNA quantitation kit, red cap. Vortex the samples thoroughly and centrifuge briefly. Add 100 microliters workflow A, 100 microliters workflow B, 110 microliters workflow C, and 110 microliters workflow D into each tube. Then close the cap. Vortex each tube thoroughly, then centrifuge briefly. Incubate at 37 degrees centigrade for 2 minutes in block heater. After that, place the tubes in the magnetic stand. Insert the plate, then let the tubes stand until the solution is clear. Reset the block heater to 80 degrees centigrade for the elution step. When the solution is clear, open the cap. Without disturbing the magnetic beads, remove the supernatant by pipette or by vacuum pump. And add 20 microliters of proteinase K and 1000 microliters of wash solution to the tubes. Then close the cap. Vortex the tubes and centrifuge briefly. Then put the tubes into the magnetic stand and let the tubes stand until the solution is clear. Open the cap. Without disturbing the magnetic beads, remove the supernatant by pipette or by vacuum pump. In this step, be sure to remove all liquid from the tube wall and tube cap without any residue. Add 100 microliters of elution buffer to each tube. Then close the caps. Remove the magnetic plate from the magnetic stand. Vortex the tubes and centrifuge the tubes briefly. Then incubate the tubes at 80 degrees centigrade for 5 minutes. After that, place the tubes in the magnetic stand. Then let the tubes stand for 1 minute. Without disturbing the magnetic beads, transfer the liquid containing the extracted nucleic acid to the new non-stick microcentrifuge tubes. The nucleic acid can be used for subsequent qPCR amplification. At this point, the sample preparation experiment is complete.